Mom jeans, corduroy, scrunchies, a lot of recent trends are outdated, backwards, and kinda retro, which is actually where retroviruses get their name.
Retroviruses are a family of enveloped viruses containing linear single-stranded RNA. Their distinctive feature is an enzyme called reverse transcriptase, also called RT. RT catalyzes the transcription of retrovirus RNA into DNA. This goes against the central dogma of biology, which states that DNA must be transcribed into RNA. Reverse transcriptase works backwards, hence its name. As with many scenarios in the world of biology, retrovirus' structure and function are closely intertwined. The evolutionary goal of life on Earth is to preserve genetic material through replication and reproduction, but viruses require a host to help carry out this process. Now, how do retroviruses utilize their envelope structure and reverse transcriptase to reproduce? They first enter the host cell through the attachment of their surface glycoproteins to specific plasma membrane receptors, which allows the virus and host cell membranes to fuse. The structure of glycoproteins has evolved to closely resemble ligand molecules that bind to surface receptors, allowing the virus to infiltrate the host cell. Viral RNA and proteins are then released into the host cell. In the cytoplasm, RT catalyzes the synthesis of complementary DNA using RNA as the template, a process called reverse transcription. RT then catalyzes the synthesis of a second DNA strand, complementary to the first. The double-stranded DNA, called a provirus, is incorporated into the host cell's DNA. Proviral genes are transcribed into mRNA by the host in normal DNA replication. Some of this mRNA is used as the genetic material for the next generation of viruses, and the rest is translated into viral proteins such as envelope glycoproteins or RT itself. The viral RNA and proteins are then transported to the cell's plasma membrane, where they are assembled into a new generation of viruses that can bud off and spread to other cells. RT allows retroviruses to infect the host cells, whose resources they need to survive, thus achieving their goal of reproduction. Several retroviruses cause disease that can be spread among human populations, like the human immunodeficiency virus that causes AIDS. RT has also been extracted for use in a laboratory procedure called reverse transcription polymerase chain reaction, or RT-PCR. RT-PCR has many applications, including identifying small amounts of circulating cancer cells and testing humans for COVID-19 by detecting COVID RNA.
So the next time your friend complains about an old trend that should never come back into style, keep in mind that its seemingly backwards nature can also be kind of retro.